Hazel eyes can often be confused with amber eyes, for instance, although they are two separate colours. Although hazel eyes may contain specks of amber and gold, they usually tend to have many other colours.
For decades, scientists believed the genetics of eye color had already been solved. Brown eyes were ancient, blue eyes traced to a single mutation, and everything else seemed like minor variation. But when ancient DNA from the Middle East was finally decoded, that tidy explanation began to unravel. For most of the 20th century, geneticists taught a surprisingly tidy explanation for human eye color. Brown was dominant, blue was recessive, and the inheritance pattern appeared simple enough that a classroom chart could predict a child's eyes with reasonable confidence. According to this model, nearly every shade outside deep brown ultimately traced back to a single mutation that altered how pigment formed inside the iris. Researchers eventually identified the molecular mechanism behind that shift. A regulatory change in a gene called HERK2 affected the activity of a neighboring gene known as OCA2, which controls how much melanin the iris produces. When OCA2 functions normally, the front layer of the iris becomes saturated with dark pigment, and the eye appears brown. When the regulatory switch suppresses that process, melanin levels drop, and the iris scatters light in a way that produces blue coloration. In 2008, a landmark study led by Professor Hans Eyberg at the University of Copenhagen reinforced this narrative by showing that blue-eyed individuals shared the same genetic haplotype. The evidence suggested that a single ancestor living near the Black Sea several thousand years ago carried the mutation that eventually spread across Europe. Within that framework, other eye colors were treated as minor variations that emerged when dominant and recessive traits mixed in complex ways. For many years, that explanation seemed convincing, partly because the data supporting it came from some of the largest genetic surveys ever conducted on pigmentation. Most of those studies drew heavily from Northern European populations, where blue eyes were common and brown eyes provided a convenient comparison group. When researchers categorized eye colors within these datasets, they often simplified the spectrum into broad bins, grouping hazel, amber, and green together as intermediate variations rather than distinct traits worthy of separate investigation. That classification quietly concealed an important geographic pattern. When eye color statistics are examined across the globe, hazel eyes appear in roughly 5% of the world's population. Yet, in several countries across the Middle East, the numbers rise dramatically beyond that baseline. Surveys in Lebanon, for instance, frequently estimate hazel frequencies between 15 and 25%, while regions of Turkey, Iran, and Syria show similarly elevated proportions compared with the global average. Plot those numbers on a map, and a striking concentration begins to appear. The highest frequencies cluster along a broad arc stretching from the eastern Mediterranean coast through northern Mesopotamia and toward the foothills of the Zagros Mountains. This corridor has long been known to archaeologists as one of the great crossroads of ancient humanity, a place where early farming cultures, migrating pastoralists, and expanding trade networks repeatedly overlapped. Despite this pattern, few early genetic studies examined the region closely because ancient DNA from the Levant and surrounding territories remained frustratingly scarce for decades. In the limestone hills of northern Israel, not far from the modern town of Piki'in, a quiet archaeological site eventually provided the missing piece of the puzzle. In 1995, excavations led by archaeologist Dina Shalem uncovered the entrance to a sealed underground chamber that had remained hidden for thousands of years behind a natural rock barrier. When researchers began carefully clearing the interior, they realized the chamber contained one of the largest Chalcolithic burial assemblages ever discovered in the Levant. Roughly 600 individuals had been interred there, around 4,500 BCE, arranged in carefully prepared deposits that suggested a tightly organized community with elaborate funerary customs. Among the most striking artifacts were tall ceramic ossuaries sculpted with stylized human faces. Their exaggerated eyes and prominent noses gave the vessels an eerie presence, as if they were watching over the remains placed inside them. The craftsmanship displayed techniques rarely seen elsewhere in the region, including metal objects formed from copper sources traced to Anatolia and the Caucasus. For archaeologists, the material culture presented a profound mystery. The objects did not resemble those produced by earlier Levantine societies, yet they also differed from traditions that emerged later. It appeared that a distinct population had flourished briefly in the region before fading from the historical record. For decades, the central question remained unresolved. Who were the people buried in Peki'in Cave, and where had they come from before arriving there? For more than 2 decades, that question lingered because the archaeological record alone could not reveal the biological identity of the people buried in the cave. The turning point arrived in 2018 when a team led by geneticist Yosef Lazaridis and Professor David Reich at Harvard University launched a project to extract ancient DNA from the Peki'in remains. Working alongside researchers from Tel Aviv University, including Dr. Hilla May and Professor Israel Hershkovitz, the group began the delicate process of sampling skeletal material preserved within the burial chamber. Scientists ground small amounts of bone into powder and used advanced sequencing techniques to isolate fragments of genetic material that had survived for more than 6,000 years. The conditions inside the cave proved unusually favorable for preservation because its cool limestone environment had shielded the bones from moisture and microbial decay. Out of 48 sampled individuals, researchers managed to reconstruct 22 complete genomes with remarkable clarity. The results transformed the archaeological mystery into a genetic narrative. The individuals buried at Peki'in were not descended from a single ancestral population. Instead, their genomes revealed a striking mixture of lineages. Roughly 57% of their ancestry traced to Levantine Neolithic communities descended from Natufian hunter-gatherers who had lived in the region for millennia. Another 26% matched early farming populations from Anatolia, while approximately 17% aligned with Chalcolithic groups from the Zagros Mountains of present-day Iran. These findings demonstrated that 3 genetically distinct populations had converged in one community during a remarkably brief period of human history. When researchers examined the pigmentation genes within those ancient genomes, the story deepened in an unexpected direction. Among the individuals buried in Peki'in Cave, nearly half carried the allele associated with lighter eye pigmentation that earlier studies had linked primarily to European populations. The variant arrived with migrants from the north, yet the genetic pattern suggested that blue eyes alone did not explain the diversity appearing in this community. Hazel eyes emerged through a different biological pathway than the mutation that produces blue eyes. Instead of completely suppressing melanin production in the iris, hazel pigmentation forms when the amount of melanin is reduced only moderately. This balance allows pigment to remain concentrated near the pupil while becoming thinner toward the outer ring of the iris. The result is a layered structure that interacts with incoming light in complex ways. One important genetic factor involved in this process is a missense mutation, known as 1,800,407 rupees, located within exon 13 of the OCA2 gene. This small change alters the amino acid sequence that influences how pigment develops inside iris cells. Unlike the blue eye regulatory switch, which dramatically lowers melanin levels, the 1,800,407 rupees variant subtly dims the pigment pathway. The iris therefore retains enough melanin to remain brown at its core, while allowing scattered wavelengths of light to interact with the surrounding tissue. As light passes through these zones of varying pigment density, shorter wavelengths scatter back toward the observer. The mixture of residual amber pigment with scattered blue light creates the shifting tones recognized as hazel, where the same eye may appear green, gold, or brown depending on lighting conditions and viewing angle. What surprised evolutionary geneticists most was not merely how hazel eyes formed, but how little evidence existed that natural selection ever favored them. Traits that provide survival advantages often leave strong genetic signatures. Patterns that indicate a mutation spread rapidly because it improved reproduction or adaptation. The famous blue eye haplotype, for example, shows clear signs of positive selection within European populations, suggesting that environmental factors or social preferences encouraged its expansion over thousands of years. Hazel-associated variants, by contrast, display no comparable evolutionary signal. Genetic surveys examining pigmentation markers across multiple continents reveal that the 1,800,407 rupees allele does not cluster within long stretches of identical DNA that normally indicate selective pressure. Instead, It appears scattered through populations in ways that resemble neutral drift rather than adaptive advantage. In other words, the trait persisted without being actively favored by the environment. Evidence for this interpretation emerged in part from research conducted at Yale University, where scientists analyzed more than 3,400 individuals representing 72 global populations. By comparing 21 single nucleotide polymorphisms within the OCA2 and HERC2 genomic region, the study demonstrated that blue eye variants produce strong patterns of extended haplotype homozygosity, while Hazel-related markers did not reach statistical significance. These findings imply that Hazel eyes endured primarily because the populations that carried them remained genetically intertwined through centuries of migration and cultural exchange. Today, the legacy of those ancient encounters can still be seen in the shifting colors of living eyes across the eastern Mediterranean and the Near East. Walk through neighborhoods in Beirut, Istanbul, or northern Iran, and the changing light occasionally reveals irises that move between green, amber, and brown as people turn their heads. The effect is not produced by green pigment because human eyes contain none, but by layers of melanin arranged in patterns shaped thousands of years ago. Those patterns reflect the moment when Anatolian farmers, Zagros highland communities, and long-established Levantine populations shared landscapes, traded technologies, and eventually blended their ancestry. Each migration left small genetic traces that accumulated inside the iris, creating a subtle optical fingerprint of ancient population mixing. In that sense, Hazel eyes function as a living archive of prehistoric movement across the Middle East. Long after pottery styles disappeared and languages shifted across empires, the evidence of those encounters still appears in something as ordinary as the color of a stranger's gaze. Hazel eyes carry no evolutionary advantage, yet they endured through 6,000 years of migrations, empires, and cultural upheavals across the Middle East. Sometimes, human history survives in unexpected places, quietly preserved in living biology. If this story intrigued you, subscribe for more discoveries hidden within our shared past.